So today we're going to talk about cell transport. Now remember, with the cell membrane, it is considered selectively permeable. That means that some things can move into and out of the cell while others cannot. And that's often dictated by whether or not the molecules have a charge, whether or not they're too large, whether or not they are polar, and therefore can't get through that nonpolar middle of the membrane. And the cell membrane is key to maintaining homeostasis, because if we don't get those nutrients that our cells need in, the cell's going to die. Conversely, you can't get the products that the cell is making out of the cell. It could have detrimental impact. There are two types of basic transport that we're going to talk about through the cell membrane. And I've given you two pictures here to illustrate the major differences. Passive, you can see this guy right here. He doesn't have a lot of energy, no energy at all. As a matter of fact, he's just chilling, laying on the couch and getting chunky. Whereas the active transport, you can see the girl on the right is active and has energy. So keep those two pictures in your mind as we go through this process. So we're going to start with passive transport. And again, if it is a passive type of transport, that means your cells do not have to put energy into the process. It will happen naturally all by itself. And there's three basic examples: simple diffusion, facilitated diffusion, and osmosis. So let's take a look at simple diffusion first. When we talk about these movement of molecules, one of the things we need to address is the concentration gradient. And a concentration gradient is just a difference in the concentration of a solute in solution on one side of a membrane versus the other, or in one particular area versus another. So for simple diffusion, molecules are going to naturally move from an area of high concentration to an area of low concentration. And again, no energy input is needed.
Into a petri dish full of water. This is to show diffusion in action. With diffusion, the particles of food coloring move from an area of higher concentration to an area of lower concentration over time. In the beaker to the left is hot tap water, and the beaker on the right is cold tap water. You can see how temperature affects the rate of diffusion. With higher temperatures, the rate of diffusion is higher, and lower temperatures, the rate of diffusion is lower.
Now let's say that we put a membrane into those beakers and the membrane is selectively permeable. Well, the same thing happens as where you just drop it into a beaker of water without a membrane. Wherever the concentration is highest, it's going to move to the area that is lowest. So it will move from high to low, and that process continues. It does not stop, but it continues back and forth at an even rate once they're equal on both sides of the membrane. So now we've talked about simple diffusion. What about facilitated diffusion? Well, whenever you facilitate something, that means you help it along. And so with this type of diffusion, there's a helper involved. No energy is needed at all, but the helper in this case is a membrane protein.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient.
Now notice on the outside of the cell, there's 4 molecules, whatever they may be, and on the inside there's 2. So the molecules are moving from high to low, but they're having to use a helper protein to do it.
And this movement is carried out without any input of energy. However, in facilitated diffusion, The movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport the molecules out of the cell.
All right, so that leads us to osmosis, which is an example of facilitated diffusion because there are little helper proteins that move, in this case, water. So osmosis will always be associated with the movement of water molecules from high concentration to low concentration. And no energy is required, so it's still a passive process. We're going to focus on different types of solutions that could impact how our body functions. So let's talk about hypertonic solutions. Now, when you have a hypertonic solution, the concentration of water inside of the cell is greater than outside of the cell. So the water wants to move from an area inside the cell to the outside of the cell. Now, this can be really damaging to cells. So, if, for example, I gave you a picture of an animal and plant cell, as that water moves out, you can see that the cell is actually shriveling up, and that's called plasmolysis. Now, the way I try to remember hypertonic solutions that the cells get skinny and shrivel up, I just imagine a hyper person as being super energetic and skinny. And so that's how I remember in my mind, oh, yeah, okay, so hypertonic solution, the cell shrivels up and gets skinny. Now, opposite of that is hypotonic. And this is when the concentration of water outside the cell is higher than it is inside the cell. And so water will naturally move into the cell. This can be a really bad thing. So if the water concentration in our blood, for example, gets too high, it will actually push into our cells and can cause our cells to burst. That is not good. However, in plants, because they have a cell wall, it creates something called turgor pressure, which means that the cells just expand and get larger, but only to a certain point. Now, the way that I remember hypotonic solutions is I think hypo-hippo. I think of a hippo being big and fat, which is what would happen to the cells if they were placed in a hypotonic solution. Now, isotonic solutions, these are the ideal. And this is where water concentration and solute concentration is the same both inside and outside of the cell. And so water will move in equal portions into and out of the cell through the membrane. All right, let's talk about active transport. So think of this as being the athlete versus the guy laying on the couch. So active transport is going to require energy. And our energy molecule in our cell is what? I hope you answered ATP, because that would be right. Now, the reason we need energy for this process is because we are moving molecules from a lower concentration to a higher concentration. And there's two types that we're going to address: molecular transport and bulk transport. So with molecular transport, you've got a membrane pump, one of those integral proteins that we saw when we learned about the cell membrane. This is the sodium-potassium pump, which is an integral pump in creating muscle contraction and nerve impulse. But what happens is that sodium and potassium are moved against their concentration gradient. So you can see here, there's one sodium ion, but it's being moved from a lower concentration to a higher concentration. There's more sodium outside of the cell, whereas the potassium is pumped against its concentration gradient from outside the cell to inside the cell. And here's our little molecule of ATP. Bulk transport is just what it implies. We're moving things in bulk. Think about when you go to Costco or to Sam's, you buy things in bulk, so in larger quantities. And we can divide this into two main types. One of them is called endocytosis. There's two types. Phagocytosis, which is right here, is the cell eating. Penocytosis is cell drinking. And so we will move items into the cell, either solid or fluid in nature. And you can see what happens is the cell membrane extends around the molecule or particle that it's bringing in and actually forms a vesicle around it and brings it into the cell. Again, this requires energy. Now, exocytosis is the opposite of that. This would be moving molecules out of the cell. So let's say we're talking about secreting mucus from a glandular cell. So the mucus is created and it's stored within a secretory vesicle, and that secretory vesicle moves towards the membrane, fuses with it, and then opens up to the outside of the cell, excreting it outside of the cell.